So we actually have a few more questions that we need to answer for this to make perfect sense. Because you might have an objection, which is, if we keep pushing glucose into the cell, the glucose concentration in here is going to get really high, and that's going to make it harder to get more glucose in. But luckily we have glucose channels on this side, on the basolateral side, that let glucose diffuse out. Its concentration is quite high in the cell because of the transporter, and so it diffuses out. You have something very similar for amino acids, and even something similar for potassium. Because if this pump keeps running all day long, it's just going to put a ton of potassium into the cell. So potassium kind of circulates, it comes in with the pump, then goes back out again, and then gets pumped right back in. So that explains how all of these things, sodium, glucose, amino acids, are crossing both membranes. But what happens to them once they get here? Do they just build up in the renal interstitium here? Well, that wouldn't be very helpful. We really want to get them back into systemic circulation. And so what they actually do is they go into the peritubular capillaries. The pressure in these peritubular capillaries is pretty low, which makes it easier for fluid to get in. And then there's also proteins here in the peritubular capillaries that exert an oncotic pressure, which helps drag all of this fluid and sodium and glucose, amino acids, into the bloodstream. Now, one last thing, sometimes people want you to know the names of some of these transporters, and two in particular. One is this sodium glucose co-transporter. It's called SGLT, that's for sodium glucose transporter. And there are actually two, and they're both here in the kidney, 1 and 2. So SGLT1 and SGLT2. And then the thing that lets glucose diffuse out of the basolateral side is called a glucose transporter. So it's GLUT, and it happens to be number 2. So glucose transporter number 2. So sodium diffuses across the apical membrane because of the low concentration inside the cell. And the transporters that it diffuses across are really interesting. There are basically 3 different types. The first is a sodium glucose co-transporter. And the way that this works is that it's a channel sitting here in the apical membrane that only lets through sodium when it's accompanied by a glucose. So if we have a sodium atom there and a glucose molecule, if they together join, they can get through this transporter. Now, of course, the same thing could happen if you had a sodium and a glucose on this side. They could join together and cross that transporter. But it's almost entirely going to go in this direction because the concentration of sodium is high out here and really low in here. So even though there might be glucose in this cell, it's going to be really hard for the glucose to find a sodium buddy to join him and to cross that channel together. And so what you'll notice is that this transporter is allowing us to take up sodium by diffusion, but also glucose. And glucose is something that we want to keep. It's how we get our energy. It's also something that we want to reabsorb from the filtrate. So with this brilliant transporter, the kidney is able to reabsorb 2 of the things that it needs. So that's the first of the transporters. And the second one is actually pretty similar. The second is called a sodium amino acid co-transporter. And so as you might guess, it's a very similar concept. You have a sodium atom and an amino acid molecule, and when they join together, they can cross the membrane. Once again, it could happen in the reverse, but because you have so little sodium in here, it's all getting pumped rapidly across the basolateral membrane. It's rarely going to happen in reverse. And so the third transporter that gets sodium across this apical membrane, I'll draw it down here because it's getting a little crowded up there, is a sodium hydrogen exchanger. So sodium comes into the cell and hydrogen is pumped out of the cell. And again, it could operate in the other direction. You could have sodium going out and hydrogen coming in, but because of the very low sodium concentration in the cell, it's going to operate mostly in this direction. So to summarize what's going on here, we have this ATPase acting as our little motor here, pumping all this sodium out of these epithelial cells. And the low concentration that creates in these cells drives the uptake of sodium as well as glucose and amino acids. And it causes hydrogen ions to be put out into the urine, but don't worry about the significance of that for now. So this is the main way that sodium gets transported across this basolateral membrane. So now we want to know, well, how does the sodium cross the apical membrane? So if you were an alien looking at this and trying to think about how it could work, you might say, okay, well, you've got one pump here on this side to push sodium out, so maybe why don't we just put another pump on this side to pump sodium across that membrane? But that's actually not the best way. And the reason is that this pump burns energy. And so if we put another pump here, it's going to burn even more energy, and we want to conserve our energy. And it turns out that we actually don't need to use up any more energy to get sodium across this first membrane. Now why is that? The reason is, because we're pumping all our sodium across the basolateral membrane, what do you think the concentration inside the cell is going to be of sodium? Well, it's going to be really low. We're going to have very low sodium concentration. And therefore, all we really need actually are channels here that don't use any energy, but that just allow sodium to go through from one side to the other. Because your sodium concentration is going to be really low inside the cell, and it's going to be normal out here in the lumen. So you'll have a whole bunch of sodium atoms out here, and very few in here. And so just by sheer chance, you're going to have more sodium atoms from this side accidentally bumping into this transporter, and therefore crossing the cell in that direction, than you will going in the opposite direction. So basically, the sodium atoms will just cross into the cell by diffusion. You don't need any active transport. Now these channels here that let sodium through are actually not that simple. They're not just sodium channels. They're actually more interesting. We'll talk about them in the next video. But sodium does just cross this apical membrane by diffusion through special channels. And that process we don't call active transport, we call it facilitated diffusion. And it's facilitated because we have those special channels that allow sodium through. And it's just a process of diffusion where sodium just goes from higher concentration to lower concentration. So we've come a long way, and it's finally time to talk about how sodium is reabsorbed. And we're going to do that initially for this first part of the nephron, the proximal convoluted tubule, where most sodium is reabsorbed. So here is the inside of the proximal convoluted tubule. So filtrate is here, and we're trying to reabsorb sodium across the walls of that. So first things first, what are these walls made of? Well, like everything else in our body, they're made of cells. And so I'm going to draw these cells nice and big. These are the cells lining the proximal tubule. And these are epithelial cells, where this is what we call the apical side. And this is what we call the basolateral side. So sodium is coming down through here, and we want to reabsorb it. And so what you'll notice is that reabsorption is actually a two-step process. You first need to get the sodium across this first membrane, and then you need to get it across that second membrane. So we'll actually start by talking about how it crosses this basolateral membrane. And the way it does that is using a very famous transporter called the sodium-potassium ATPase. And so that's going to push sodium out and take potassium in. In particular, it puts out 3 sodium and it takes in 2 potassium. Now this thing uses up ATP. ATP is our source of energy that our body uses. And so this transporter requires energy, which is why we call this active transport. So active transport burns energy.